This module is an introduction to some of the basics of flow cytometry instruments. My name is Christopher Lazarski, and I am the Flow Core Director at Children's National Hospital. This module will help explain how cytometers collect data and how users should set up their instruments in order to recover readable quality data, which they can then use for publication. Terms which will be discussed include wavelength, excitation, and emission, as well as filter sets and corresponding gains, along with the spectrum from fluorescence markers. Topics which will be discussed include identifying fluorochromes, setting your gains, and troubleshooting problems which you may encounter when collecting your samples. Part of flow cytometry, like all science, has jargon associated with it. One of the terms includes staining our cells or staining your cells. This is because fluorochromes are often referred to in different colors, such as FTSE or PE, and therefore we are staining our cells with different colors. In general, flow cytometry experiments involve measuring the amount of fluorescence which is being emitted by cells in suspension and loaded on the machine. We use antibodies specific for surface markers or intracellular proteins to help phenotype the cell population. In this case, for example, do we have T cells in our population? How many T cells do we have? Or do we have B cells? Are they activated? These antibodies, which are bound to the surface markers, have fluorescent molecules attached to them. In this case, for the T cell marker, this has a PE fluorescent molecule attached to it, and this fluoresces in the PE channel, while we have a second antibody which targets a B cell surface marker, such as CD20. This second antibody has a red fluorescent marker attached to it, APC. This fluoresces in the APC channel. Flow cytometry instruments are compatible with both antibodies, which have fluorescent markers, as well as with fluorescent proteins. Some of these proteins could be GFP or mCherry, as well as with fluorescent DNA dyes, including propydium iodide, 7-AAD, or DRAC5. Our instruments then record the frequency of these marked or stained cells and the intensity of the staining signal using detectors specific for that fluorescent protein. The flow part of flow cytometry refers to the fluidics which the instrument uses in order to funnel the cells past the lasers and detectors in a single file line. Lasers excite fluorochromes on the labeled cells, which then release this energy as light. This light is emitted across certain wavelengths. The emitted light is redirected using various filter sets to be collected by specific detectors. The detectors then convert the photons of light to an electric signal. This signal is displayed numerically as the intensity of fluorescence. Cytometers record thousands of events per second in all channels. This is another unique feature as compared to a microscope. As the cells pass by the lasers in order, each laser bombards the cell with light, and the instrument records the signal in all channels. So in this example, when a cell stained with a red fluorochrome APC passes through the instrument records data in all the channels, including the violet channels, the blue channels, and the yellow channels, along with the red. This is important if the cell has background fluorescent intensity in some of these additional channels. Each instrument has its own set of lasers and detectors, and when we refer to channels and colors, we are referring to the detectors which the instrument records the data with. Each detector then records the light intensity using a filter with a specific wavelength range. Some fluorochromes will work better on specific instruments than other instruments because they have a laser which activates the fluorochrome better. The prime example for this are red fluorescent proteins, such as mCherry or tomato and RFP. These are activated much more efficiently by an instrument with a yellow-green laser as compared to a blue laser. While the blue laser can activate the red fluorescent proteins, it is an inefficient activation, less than 20% of the intensity as compared to a yellow laser activating the red fluorescent protein. Please confirm that your experiment is compatible with the instrument which you are using. In order to understand the filter sets, a detector is essentially a window. The center of the window is the first number. For FTSE on our Cytoflex Oscar, the center of the window is at 525 nanometers. The width of the window is the second number after the slash. In this case, it is 40 nanometers. This is the total width of the detector window. So if the center is at 525, the width is 40, this would be plus and minus 20 nanometers on either side. So the FTSE channel on Oscar therefore has a range of 505 nanometers to 545 nanometers. Manufacturers will publish both the excitation and emission wavelengths of fluorescent proteins, and our core has a listing of the instruments and the filter sets on them in order to compare whether your fluorochrome is compatible. At Children's National, our current set of research equipment includes 4 instruments at the main hospital. We have 1 sorter at the main hospital for enriching rare cells and populations of interest for later downstream analysis, such as sequencing or single-cell cloning. The sorter has 4 lasers, including a yellow-green laser, and 15 detectors. We also have 3 analyzers at the main hospital, 2 of which are traditional flow cytometry analyzers, one which has 4 lasers, Cookie, a yellow-green laser, and 13 detectors, and a second Cytoflex instrument, Oscar, which has 3 lasers and 13 detectors. We also have a Cytec Aurora analyzer, which has 5 lasers, including a UV laser, and 64 specialized detectors. This is a spectral flow cytometry instrument, and for that, this requires special training and a separate module on spectral flow cytometry. At the Research and Innovation Campus, we have 2 instruments. We have another Cytoflex sorter. This sorter has 3 lasers, a red, a blue, and a violet laser, and 10 detectors off of those 3 lasers. We also have an older Miltenny MaxQuant, in this case FaxQuant Freddy. This analyzer has 3 lasers and 8 detectors off of those 3 lasers. All the analyzers can be user-operated after training on the use of the software. The 2 sorters are reserved for core staff operation because of the technical hurdles in setting up the sorting instruments. Moving on in our understanding of jargon and flow cytometry, fluorochromes are proteins which can be stimulated by lasers to emit light across a defined wavelength. Fluorochrome excitation is the wavelength of light which excites the electrons into a higher energy state. These electrons quickly collapse back into a less energetic state. This releases a little bit of energy as heat and the rest of the energy as light. This light is shifted relative to the incoming light to a longer wavelength, which is then read out by the machine. A list of fluorochromes compatible with the specific channels and with our instruments is discussed in our panel design module. We also have a link on the website to specific channels and corresponding fluorochromes provided here. Besides the lasers and the detectors on an instrument, the gains have the most direct effect on the data which will be collected. The gain is a setting to increase the signal intensity read out by a fluorescence detector. It doesn't affect the power of the laser. When you change the gain, you're not increasing the laser power, nor does it change the number of photons that a cell is emitting. Rather, the gain is a multiplier which has been applied to the signal coming from the detector. As a user, you can imagine that increasing the gain on our instruments is similar to turning up a guitar amplifier to make the music louder. Voltage was an older term. It was originally referring to increasing the background current applied to a different type of detector. For our use, voltages and gains are interchangeable. Our instruments, as an aside, use avalanche photodiodes, which are basically semiconductor versions of the older style photon multiplier tubes. One point of emphasis, which we repeat across several of the modules is that gains have the most direct effect on your flow data. Increasing the gain will increase both your signal and your background. It is important for a user when setting up their instrument to select gains which are appropriate for their cells and their fluorochromes. This is based on the background of the cells and based on the signal of the fluorochrome which they are investigating. Gains affect both the primary signal intensity and the spillover and compensation for multicolor experiments. Importantly, there is a limit where increasing gains past this certain amount does not change the relative signal of your positive marker above the background. This difference between the signal and the background can often be identified as a staining index. Therefore, beyond a certain gain, the staining index does not change. Recommended gains are set during QC weekly and are a starting point for users of the Cytoflex instruments. These recommended gains have been programmed by the manufacturer to be the minimum gain needed to see a dim fluorescent marker above the background. Data has been collected for our two Cytoflex instruments, which identifies the minimum and maximum gains in order to clearly distinguish signal from noise. One of the questions which may intimidate users of the Cytoflex instruments is deciding why and when should they change their gains and how much should they change it. Our recommendation in the flow core is that best practice would be to start from the weekly QC recommended gains. This is because our cytometers have a large dynamic range from 1 all the way out to 10 to the 7th, and often the recommended gains will work for the majority of your cell experiments. When you don't see your cells, often what is happening is that instead of needing to increase the gain of the size or forward scatter and the granularity or the side scatter, what is happening is that the cell is simply off the scale. It is simply not in view, and the user can just adjust the scale of your forward and side scatter graph in order to bring the population back into view. So we would recommend first change the window scale and not necessarily the gains for forward scatter and side scatter. After changing the scale, then see if you can locate your cells. If not, then you can start to consider changing the forward and side scatter gains. Another situation where you might want to change your gains is when your fluorescence signal is too high or too low. This is because the software cannot recover data which has already been pushed up against one of the axes. A situation where your fluorescence signal might be too high would include a lentiviral transduced GFP, can often be very high, or some cell labeling proteins such as calcine dye or some of the CFSE vital dyes can often have a very bright signal. In this instance, then, you would reduce the fluorescent detector gain for the channel that you're looking at. For GFP, that would be FITSI. Users may also change the gains if there is too much spillover between channels which are close to each other. In this example, when we have spillover between our close channels, such as KO525 and BV610, we increase the gain of one of the channels to help distinguish it from the spillover staining of another channel. Also, a signal cannot be rescued if it falls back into the negative population. In this situation, the data is the data. This is why, again, we recommend starting from the QC recommended gains and moving from there. Finally, another situation is that bigger cells with more cytoplasm will have more background. Cultured cells and tumor cells often are larger and definitely have more cytoplasm than primary lymphocytes or peripheral blood. This is why users would need to collect their data on the background intensity of their specific cells and not rely on assumptions for the intensity of their cells. In order to make a cytometer work the best for your experiment, we would advise that users would take the time to learn the basics of the instrument. This includes the startup, shutdown, cleaning procedure, as well as basics for creating an experiment, collecting their samples, and then collecting their data. By becoming familiar with the configuration of the cytometer, you would understand which fluorochromes fit into the channels and how large your panel can be, and then still work on the instrument. Additionally, start from recommended gains. Be conservative in the changes that you would make to the gains on the machine. Our Cytoflex instruments support creating templates from experiments that have worked before, and I would recommend to use this option. This will preserve the gates, the settings, as well as any compensation. And in this way, you can be sure that you are collecting your data with gains and settings that have previously worked. We would also recommend that users give yourself enough time when setting up and performing your experiment. This can often be the case where prepping the cells, staining the cells, and washing the cells takes more time than planned, and users may end up being or feeling rushed when they arrive at the machine. Please abide by the calendar. Our current calendars are shared calendars under Outlook, and we have many users of the machines who all need to get their experiments done. Assume at least 2 to 3 minutes collection per sample. Also, if possible, please pass your cells over a cell strainer in order to remove cell clumps and debris which may clog the machine. Ask for help from the flow cytometry staff if you are unsure, and we can both advise and provide assistance to setting up your instrument and collecting your data. In summary, for understanding cytometers, cytometers collect light emitted from fluorescent markers bound to cells, and each cytometer has its own set of lasers and detectors. By adjusting your gains, these adjust how the detectors measure the intensity of light from your cells. And importantly, users should take the time to measure the background intensity of their specific cells using their specific antibodies to ensure the best result for collecting your experiment.